Maybe hello and welcome, everybody. Maybe wait just one more minute in case there are some people still joining us before we officially start. That's okay with you. Okay, maybe we can start already. So, hello again and welcome. I'm Christina. I'm happy to be your host again for this training series dedicated to turning biodiversity observations into insights. Um, the 1 second. Okay. So I will be your host for the next 90 minutes. Hopefully, you'll be engaged and stay with us up to the end. Today, we're going to learn about occurrence cubes and how to use them in the field of biological invasions. We have another two amazing trainers with us today, Maarten Trekels and Sandra MacFadyen. There will be a short break in between the presentations. There will be some hands-on sessions and also Q&A sessions. Please use the Q&A section to address your questions, and we will try our best to answer them as soon as possible in real time. And then we'll just part ways, and hopefully you'll join us again next week. In case this is your first time joining us, please allow me to take a couple of minutes just to introduce the b-cubed project funded by the Horizon Europe Research and Innovation Program. The training series is organized in the framework of this b-cubed project, who's aiming at improving the integration of data on biodiversity and also at standardizing access to biodiversity data. You can find more information about the aims of the project and the outputs of the project on the project website, which is b-cubed.eu. And we have with us today our coordinator, our brilliant leader, Dr. Quentin Groom from Meise Botanic Garden, Belgium. He is coordinating a large team of amazing people that are interested in biodiversity data coming from different institutions across Europe and also beyond European borders. In case you missed our first session, we talked last week about species occurrence cubes. We had an introduction, a brilliant introduction into the subject. Our trainers were Andrew Rodriguez and Lina Estupinan Suarez. If you missed it, you can find details and some training materials on the b-cubed project website under the section news for the moment. And today we are going to continue our discussion on occurrence cubes and how we can use them in the field of biological invasions. As previously mentioned, our speakers today, our trainers today, are Maarten Trekels and Sandra MacFadyen. Maarten is a biodiversity data scientist based in Meise Botanic Garden in Belgium. He's working at this interface between data science and biodiversity conservation. He is passionate about many things, among which open data, development of data standards, and also making accessible natural history collections across Europe. He is also working on plant communities during his PhD in mathematical biology at Stellenbosch University, and he's a collaborator of Sandra. Sandra, she's a postdoctoral research fellow at Stellenbosch University in South Africa. She completed a joint PhD in botany at the Stellenbosch University and landscape ecology at the Vrije University in Amsterdam. She's also passionate about many things, among which biodiversity conservation and improving the monitoring of biodiversity. And she's using different models in her research, like macro-scale ecosystem dynamics and mathematical ecology, complexity theory, many interesting ways she's approaching this biodiversity monitoring and conservation in large protected areas. I would love to visit her office one day because she has been working for almost 30 years in Kruger National Park. And today they took time from their busy schedules to talk to us about this occurrence cube and how they can be used, in the field of biological invasions. Just before I hand over to our speakers, please I'm asking now, kindly inviting the audience to answer a quick poll, three short questions, just to for us to get an idea about the participants, about the audience that's joining in these training sessions. The first question is related to the country you're based in, and the second question is related to the stage you're at in your career. And the third question is related to the sector that you activate in currently. Hopefully, we'll have a good mix of people attending, same as in our previous session. I will just take a couple of minutes for people to answer to these questions. I see that results are starting to come in. I think we have a good coverage, spatial coverage as well. For the moment, I'm not able to see the answers to the first question, but I think I will get them at the end of the session. However, I can already see the current stage in the careers of the participants. So we have with us students, which is perfect, undergraduate students, master students, and PhD candidates, and several postdoctoral researchers. And also, yes, we have a good coverage of people that are at the early stage in their careers, mid-career, and people that are more experienced, like have more than 10 years experience in the field, which is perfect. It's great. Also, a good coverage here in terms of the sector in which participants are engaged. Close to 30% academia and higher education. So we have a lot of researchers with us. Research institutes, yes. Quarter of the participants. But also, yes, I'm happy to share that we have people from the government and public administrations joining, NGOs, private sector, and consultancy. And the students, which are not currently employed in a field, but maybe they get some idea about potential careers, future careers. Okay, I think we can perhaps end the poll, maybe? Thank you. And now I will just hand over to Maarten and Sandra to share with us a bit of their experience.
Thank you very much, Christina. I will start sharing my screen. I have the difficult task to come after the excellent presentation of Andrew of last week. So I hope it will be a bit less interactive as it was before, but I will do my best to keep it interesting to everyone. So welcome everyone, and thank you for the introduction, Christina. I think we already went through this, so we are now at the 27th of February, where we're going to look at the occurrence cubes and biological invasions. We had our nice present introduction already. I will focus on a Python package, p3 alien, to track the target 6 of the GBF, while Sandra will give you an overview of an R package on invasibility. I will start with giving a general overview of the b-cubed project. We already had that, but I will go a bit deeper into the essential biodiversity variables and very briefly introduce the conventional biological diversity targets on invasive alien species, where in part two, Sandra will go into detail in the invasive mapper R package. So first, B3 Alien. So this is a Python package that was developed to report on the coming Global Biodiversity Framework Target 6.1 indicator. And why are we using b-cubed for this? Well, b-cubed, the aim of b-cubed is to, or the challenges that we are facing is to have rapid, reliable, and repeatable workflows to report on certain indicators. Moreover, there are so many information that is gathered on biodiversity at different scales, so that we have to try to improve the integration of this data on biodiversity from different sources. And b-cubed is a way to standardize the access to this biodiversity data to make sure that everyone that is using the products of b-cubed is able to report on indicators in a similar or in the same way for each country or each region that is important for us. It's also the goal is to have easy workflows that are not requiring a lot of modification by the users. You will hear in the next presentation, especially the ones that are developing the R packages, something about the B3 verse, which is an ecosystem of R packages to calculate the indicators, and they are all gathered in different steps within the data flow. So in this case, we go from left to right, where I'm now focusing on a Python package. So we are a bit different from the other ones. I focus mainly on this middle part, is the calculation of the indicator. The data processing and data generation, we will leave up to gbif to generate the cubes. And my Python package is more focused on calculating the indicator. So if you hear in one of the next sessions something about the B3 verse, B3 Alien is a bit different. So it's not part of this one, but it's a Python implementation of the calculation of an indicator. One thing I want to just highlight from the b-cubed project, and that's an important part also for the indicator that I will be presenting, is that we are looking at primary data. And this primary data, we are focusing mainly on data which is coming from gbif, that we transform into essential biodiversity variables. Basically, these essential biodiversity variables are three-dimensional, but it can be multiple dimensions as well. But the main are data structures that have a spatial component, a taxonomy component, and a time component. Using these, which can be derived or can be created from the raw primary data, you can also model these cubes according to certain algorithms. From these on, you can calculate the indicator itself. And this indicator is typically just a single value over time to see trends on biodiversity. Using these indicators, we can then inform policymakers again, but also data collectors on where potentially extra data needs to be collected, or different types of data have to be collected, and so on. So this is the main concept, and when I present the B3 Alien Python package, I will try to follow this flow a little bit so that you can see how we see this in practice. The practical implementation I'm going to show you is we are starting off from gbif data. We generate a more or less standard data cube, which already includes the aggregation of data, some filtering, some data cleaning in the process itself. And I will go over, Andrew showed this very nicely last week, but I will quickly show it again in my practical hands-on presentation as well. And then you get our data cube with a spatial component. You have the taxonomy, because this is, for example, for one taxon plotted, and the time component. These data cubes have another interesting feature, is that they are compatible with other types of data sources. And for example, if you look at remote sensing data, you could overlay these data cubes with any other kind of spatial information and aggregate the spatial information at the same level as you are calculating your biodiversity data cube. How are we going to generate this data cube? Andrew showed last week the SQL interface to GBIF. To make everything relatively easy and simple in the workflow I'm presenting today, I will focus myself only on the graphical user interface, so that after this presentation, anyone should be able to generate a data cube and run my workflow afterwards. What am I trying to calculate in my workflow? Well, that's on the CBD targets on invasive alien species. So target 6 of the Global Biodiversity Framework says that we have to reduce the introduction of invasive alien species by 50% by 2030. So in order to reach that target, or for countries to be able to report on this target, we need a way to monitor this. And for that, they defined a number of indicators, and these indicators are more or less organized in order of importance. The headline indicator, the most important indicator, is the 6.1, which is the rate of invasive alien species establishment. And it's this one that we are going to focus on the most. There are a number of other indicators that are important for this. For example, the rate of invasive species impact and rate of impact. Well, this one will be covered by Mukhtar in one of the following presentations. But you also have rate of invasive alien species spread. And this is something after it still needs a little bit of research, but which we can also monitor using the data cubes. The same with number of introduction events. Then there are some others that we could report on. Why are we going to use these data cubes to monitor invasion? Well, we have actually all the basic information that we need to monitor these invasions. For example, here you see this temporal component going from the 80s to the 20s. You have a taxonomy, so in this case it's a single species, and you have a number of species for each grid cell that is here indicated on the map. And so this is how we are going to use it. And the most important thing on my workflow is that I'm not relying on any data that you still have to collect yourself. So you don't need to go and collect your own checklist or your own data sources. You can do that if you want to, but the idea is that the basic workflow just uses GBIF and the occurrences that you can find on GBIF as a source to determine when a species enters a country, and the GRIS, the Global Register of Introduced and Invasive Species, which is these checklists are also on GBIF and contains for each country a list of invasive or potentially invasive species in a country. So it's only those two data sources that I will present and use in my workflow. Of course, the workflow can be modified to incorporate other types of data sources, but essentially in the beginning or in the most simple form, it's only assuming those two sources. How does it work? Well, both the GRIS checklist and the occurrence data are on GBIF. So from GBIF, we generate a SQL download, and we take the GRIS checklist of the country under consideration. From this SQL download, you have two routes. either you have another additional spatial information on the grid that you are using, and you can generate a GeoParquet file. The nice thing about having this GeoParquet file is that you afterwards can have nice interactive maps on what you're considering on your data cube. If you don't have any grid to align your SQL download with, that's not a problem. Also, the workflow works perfectly fine just on the CSV download and can generate you the cumulative curve of number of introductions. Then the second download that we use from gbif is the quiz checklist, where we make a checklist object from. And by combining the occurrence cube and the checklist, we can simulate the rate of establishment of invasive species. And all of this is wrapped in a single B3Alien Python package that you can install, and where you can find all kind of functions on each of the objects within this Python package. The Python package is freely and open available on GitHub with some additional information. So if you are using the package and you want to and you encounter any issue or any problem, you can actually use GitHub to track any problems that you have with it and create an issue on this. I will look at it as soon as I can and then try to fix it for your specific use case if there is a need for it. It also comes on the GitHub repository with an exemplar workflow, but for the course of this training, I will also I also created a different GitHub repository that contains all the training materials, such that you don't need to go and check the GitHub repository and download data. There is already a pre-made repository, and I will share the link later, where you can find already exemplar data in there. And so you can run this workflow for any country that you want, any country that has a GRIS checklist. And so here you see an example where I did it for 4 different countries: Belgium, South Africa, Australia, and Costa Rica, where you can see the change in the rate of establishment over the years. So for example, in the case of Australia, the black curve is the cumulative number of occurrences. You see that this curve is flattening later on, so the rate at which species are entering the country is decreasing. The reddish curve here is a simulation. So basically, there is a simulation with an error margin that gives you a number on this rate of establishment. The third element that you can see on these curves is a measure for the observation effort. So the blue curve with the dots is giving you a kind of indication on how much observation effort is done for getting or tracking species in a country. I will, when I go through the different steps in the notebook, I will go deeper into this. But you can see, for example, why this is important to have at least a qualitative understanding on how this observation effort has an effect on the curve. And if you look, for example, here in the case of South Africa, you see that when the observation effort goes up, you will also subsequently see that you record more of the introductions of invasive species, although there might be a slight delay on when these species are detected. You see that I did the calculation here for these different countries. Belgium is a bit of a special case, because in Belgium we have a more detailed GRIS checklist where you have introductions dates included, where you see that this expert curated checklist is giving us a little bit of a higher change in rate of establishment than the one calculated through my workflow. But if you look at the confidence interval, you see that essentially both are falling within the same interval. So statistically, the one that is calculating through gbif is not significantly different from the one that is done through expert curation of checklists. And what is the big advantage of using this strategy? Well, it's a simple workflow and it's readily usable for any country in the world that has a GRIS checklist. And so, for example, you can see here that I did the same workflow for all the countries that I could run the workflow up. In this top left figure, you see that when it's more bluish, the rate of establishment is going down. When it's more reddish, the rate of establishment is going up. And so I could calculate this figure for the whole globe in about half a day. So it took me half a day to calculate it for the whole globe. You do see that some of the countries where there is a data deficiency or there is no GRIS checklist available, you might see that there are missing data. So for some countries, it's not possible, or the model just fails for data deficient countries. So it's heavily relying on the GRIS checklist and on the quality of the GRIS checklist, but also on how much data for these countries is available on GBIF. So it's basically a warm invitation for any of these countries to make sure that the GRIS checklist is up to date and that there is a significant amount of monitoring done to be able to have some information on the invasive species. It's easy to integrate in existing workflows, at least in existing Python workflows, because the package can be easily installed through PyPy. And in the future, the idea is that this package, it still relies on some bootstrapping mechanism, which is a bit slow, and also having to start off from a country cube might be a little bit difficult. So in the future workflow, we will make sure that we can actually run this the same workflow for multiple countries at the same time. And also, we will look more deeply into how to report on the complementary indicators that are needed for target 6 of the CBD. Okay, this was a very general introduction and a very broad idea of what I prepared for this, what I created in the B3 Alien package. For the rest of this, I will go step by step through the workflow to show you how easy it actually is to go through it. It's maybe a bit difficult to do it simultaneously, but I would invite you to try it out yourself. And so I created a training repository on my GitHub. The link is here on the slide, which contains the full workflow, but also already contains some exemplar data that it's not too difficult for you to reproduce the workflow. Once you get used to it, you can download your own data and your own GRIS checklists to do it for your own country. So with that, I will shift to the workflow. Moreover, so let me show you, it's the package is available on GitHub, but it also comes with an extensive documentation. So basically, and I have to see because my controls are a little bit in the way. I have to move them out. Yes. So if you go to b3alien.readthedocs.io, you will actually get an overview of all the documentations of this Python package. So all the functions that are used within the software or within the workflow are explained here. And for example, if you go to the Occurrence cube class, it gives you an idea on how to create the class, but also which functions that there are available in the B3 Alien package. So besides the workflow that I'm going to show here, you will be able to do other stuff with B3 Alien in Python as well. So for example, you can aggregate counts per cell through this function. It gives you the parameters that you need to do this and what kind of output you get from this function. And all functions are explained here. If you find errors in the functions or things that are not clear, please let me know, and then I can fix this for you. This is the repository of the training. At the moment, it's a rather empty repository, but it contains two important things. So to run this workflow, the easiest to do is to clone this repository on your own computer, or you can just download it as a zip file and unzip it on your computer. And the two important files that you have to use here are the exemplar workflow Jupyter notebook and the data folder. In the data folder, there is already some pre-prepared data for you, so that it becomes easier to run the workflow. Once you have a Jupyter notebook open and you loaded the Jupyter notebook, which is here in the repository, you can start playing around with it. If you don't have Jupyter installed on your computer, which is not so difficult, but in case you don't have it, you can also upload this Jupyter notebook to Google Colab. And if you want to use Google Colab, it works fine in there as well. You only have to make sure that you change the links to the data folder then. So in case you don't have a local Jupyter notebook, please feel free to use Google Colab or any other hosted Jupyter notebook somewhere. So when you have it open, it starts like this. This is my Jupyter notebook. And it starts by installing the B3Alien package. I don't have to do it now because I have it already installed and it takes a little bit of time. So for the sake of demonstration, I will not do it again. But when you install the B3Alien, it's just pip install the package and it installs all the dependencies that are needed to run this package. This first box, I just load in the package and some other packages that I need for visualization. For the ones that never used a Jupyter notebook, either you can press on the top here the play button and then it will run this cell. Otherwise, you can also just take Shift Enter and press, and then you will see that there is a star appearing here. When it's finished, it will give you a number, and that number is the number of the cell or the number of run cells that you already have. So I pressed enter, there were no error messages, so the package is loaded. And then the next step in the workflow is to generate a biodiversity data cube from gbif. And for this, for the sake of demonstration, I just use the standard data cube that you can generate from gbif. And how do you do this? I just briefly gonna repeat what Andrew already showed you last week. You go to gbif, you go into the occurrences, and there you filter on the country that you need. For example, let's take France as a country, and you will get all the occurrences. Actually, you will see that it's sometimes easier to use one of the administrative areas because it can happen that if you just filter on a country code, there are occurrences falling out of the country. So if you use the administrative area, you go to the map view, you will see that you only have occurrences that are situated in France. So this is the occurrences that we want. So you can almost immediately go to the download. In this download, you see the different formats in which you can download your data as a simple CSV or a Darwin Core archive, but we are here interested in the cube format. So you press cube and you will get this additional screen. We are, as a taxonomic dimension, interested in the species. For the temporal dimension, actually the workflow is now designed to work on a monthly basis. So you take year and month as the temporal dimension. And as a spatial dimension, you can pick whatever grid you want. So you can take the EEA reference grid for Europe or one of the other grids that are defined here. You can even do it for the full country and look at the country as one grid cell. For the sake of demonstration, we can go for the extended quarter degree grid cells. If you take that option, you can still even choose the spatial resolution of your grid. At level 2, it's about 28 kilometers its size. The higher in level, the smaller the grid cells become. Then you can randomize points within uncertainty circle. For this workflow, it really doesn't matter. So in many cases, I just pick no. And then you can use any kind of counts at higher taxonomic level. This can be interesting, especially if you want to have a measure on observation effort. So you can leave them here or only leave the ones that you want as taxonomic level. We don't need the minimum coordinate uncertainty for this workflow, but there are some data quality parameters here. And so you can remove records with geospatial issues, remove records that are not matched to a taxon. You can leave those filters on. The one filter I remove is the remove records at country centroids, because we actually do want to see all the invasive species coming in. So even if it's mapped to the country centroid, I don't really care about this component here. So I remove that data quality flag. And then you can either press download and the workflow will work perfectly fine on the cube that you are generating here, or you can edit the SQL here. So basically all the options that I selected here, you can edit them also as in the SQL download here. This is what you have seen last week. If I want to make some edits, you can here press the start editing button and we can start changing the SQL to our needs. Why do I show this? Actually, there are some ways of getting an ID on observation effort. And one of the metrics that I was trying to use is number of distinct observers per grid cell. But this is not standard coming out of your SQL download. So what I typically do is add another column to my data cube. In this case, I want to count all the distinct recorded by, and I call this parameter distinct observers. The only thing I did now is just modified the SQL a little bit to have one additional measure on observation effort. You can make this measure as complex as you want. You can modify the SQL query as complex as you want, but it's not necessary. So basically, in whatever you do in the workflow, you don't need specifically to make it more complicated. And then you can start downloading this data cube. Like I said, I already downloaded the data cube, so we don't need it. If you look at the data folder here, this is the data folder with the pre-prepared data. I have already 2 CSV files that I renamed from gbif. There is one at level quarter degree grid cell level 2 and one at level 5. So you can load those CSVs in. Like I said, it's also working on GeoParquet files if you want to have more visualization of your data. There is an exemplar parquet file here for the level 5 data. It's not there for the level 2 data, but the exemplar workflow shows you how to do it. So if you want to create a GeoParquet file at level 2, you just give you the path to your CSV file and to a geo package that contains the geometry of your extended quarter degree grid cell in this case. If you don't know where to find it, there is a link here to a sub-page on the gbif website where you actually can find all these custom geo package files of the grid cells levels. And even for the other grids that are generated through gbif, like the EEA grid, you can find the same files here as well. So if you want to link them up to a spatial explicit file, you can look at downloads.gbif.org/grids, and you can find most of the grid cells that are used in the gbif download. So what can you do if you have those two files? You can use the toGeoParquet function within B3 Alien that just takes the two files. Depending on where you get your grid cell size, your grid geo package, you might have to check what IDs are used as cell codes. These are the standard one, but it can change for your case. And then an export path where you want this Parquet file to end up. I'm not going to run this cell because this takes a little bit longer, and for the sake of demonstration, it doesn't really bring too much to it. But if you run this cell, it will create you a Parquet file at this location. The other thing that you need for running my workflow is the GRIS checklist. In this case, it's the GRIS checklist of Portugal, which you can also just find on GBIF if you go to datasets and you search for the GRIS checklists. You can find all the GRIS checklists here. The one that you have to download is the one for Portugal in this case, but also here I already downloaded here and unzipped it in the folder. In many of the basic GRIS checklists, there is no matching to the GBIF taxonomic backbone. So there is a function added to taxon matching, which is taking all the species that are in this GRIS checklist and match it to the GBIF taxonomic backbone. Again, I'm not going to run this one here because it's making API calls to the GBIF API, which can take a while and is also stressing the GBIF infrastructure. But once you did that, you will find a file merge distribution in there, which we can load as a checklist. We can load our occurrence cube like this. And then there are some additional functions. For example, if you want to calculate the observed species richness in a cube, this is the function that you have to use. I might be going a bit fast here, but all the explanations should be in the workflow. Let's run this one. And then because we matched it up with a geopackage file and we have a geoparquet file, we can plot this richness on a map. So basically, I did it for one island on the Azores. And you see here the species richness. The darker color it is, the more species are observed in this data set. It also contains some marine species. And you see how this is distributed over the island. This gives you already an idea on where most of the occurrences are coming from. In the next step, we are going to calculate the rate of establishment. For that, we need first the cumulative number of species. And when this is finished, we can, from this cumulative number of species, we can calculate the rate. We can filter over some time. So if you want to do the simulation, we don't have to do it necessarily over the whole time range. So in this case, I'm calculating it from 1970 to 2022. Again, you can choose your filters yourself. And then using that data, I can do a simulation. So in this case, the simulation is using the Solo Costello model to get an ID on the rate of establishment. So after the simulation is finished, you will get the dark line is the discoveries, and then the dashed line is the simulation. In this case, it was only like a small island and a subset of this island group. So don't look too much on how this curve looks like right now. And then you can also get a number for this, the fitted rate. The simulation is nice, but it would be also very good to have an idea on what is the error that you make on this calculation. So there is a bootstrapping method included to do the simulation and get an error margin. For the demonstration here, I'm going to do only 100 iterations and plot it then with confidence level. And then you will see that it will start the bootstrapping. Normally with 100, it should go relatively fast. And then once the simulation is done, it will give you at least a rate of establishment or a changing rate of establishment, including the error margins on this on the calculated parameter. Okay, it's finished, and you see here the the curve, including the 95% confidence interval. To get an idea on how this relates to how much observation effort there is, we have an extra step that calculates the survey effort. So there are several ways to do it. The most simple way is to aggregate counts at a higher taxonomic level. For example, in this example here, I'm aggregating everything at the level of plants. And then I aggregate also in time, just for plotting it. I filter on the same year, and I plot the observation effort. And you see, with the onset of citizen science projects, there is a lot more observation effort happening here in the last decade. Remember that I had this more advanced, between quote, calculation of the observation effort by using the number of distinct observers. So you can actually change the calculation type to checking the number of distinct observers, which is using exactly the same way, and now plots it together with the curve that we calculated before in a logarithm, in this case, to just see it a little bit better. So now you get a measure of observation effort together with your calculated curve. Again, how is this observation effort distributed in space? Since we have the spatial component as well, you can actually look at how the observation effort is distributed in space, like you see here, which corresponds quite well with your observed species richness, because the more you observe, the more species that you will encounter. So this gives you at least an idea on observation effort over space. This is for the higher taxonomic level. We can do exactly the same for the number of distinct observers, which gives you qualitatively more or less the same image. You see slight differences between the methods that you use, but qualitatively you do get an idea on what are the places where observation effort took place. And so with that, I want to end my presentation and see if there are any pressing questions right now. Let me quickly check. Everything is answered, I think. Okay. At the end of the webinar, you will find you will find our email addresses. So if there is any other question popping up, feel free to contact us. Also, okay, this one I can answer live. Can we use it in Visual Studio? Yes, you can load the Jupyter Notebook just in Visual Studio or VS Code and run it there. The only thing you have to make sure is that your Python environment has the B-tree alien package installed. So, and if you encounter any issues with the B-tree alien package, please feel free to file a GitHub issue, and we will make sure that that I will follow up on those. And I think with that we can go for a very quick break and then switch over to Sandra's presentation. Let's say that we have 4 minutes break, and then we will reconvene here. Thank you very much. I see one more question that I can quickly answer. So a local grid and occurrences based on local data, it's possible. Basically, in the background, B-tree alien is using GeoPandas as an X-array to do the calculations. So as long as you have the standard columns or some basic information, species, number of species per grid cell, and your geometry, you should be able to modify the workflow to work with yours as well. It might involve a little bit of Python skills. Okay, so we can start again at 5 to 4 with part 2 from Sandra.
Okay. Good afternoon, everyone. I hope you can all hear me okay. You're all back from your quick body break. We can continue with part 2. As Maarten's already introduced me and Christine's already introduced me, I'm Sandra MacFadyen, and I'm going to take us through a brief introduction to a framework to visualize trait dispersion and assess species invasiveness and site invasibility. So this is through the R package in BasiMap. We're not going to have enough time to run through a tutorial on this, so I'm just going to give you a brief overview of it. Part of it does get covered later on in another session I'm doing on the 13th, Friday the 13th, so we can go through some more hands-on stuff then. But for now, I'm just going to take you through a quick overview of the package and the invasibility cube itself. So as Maarten has already shown you this slide, it's essentially just giving you a rundown of the B3 verse, which is the different packages and the different workflows that have been developed through the b-cubed program. The one that we're going to be focusing on now that I'm going to show you is the BasiMap R system, which is the R package that looks at invasion fitness. There is a website with a bunch of tutorials on that you can follow it. And there's a step-by-step instruction there using examples of butterflies in South Africa. What I'm going to run you through now is an example of some parquet data that Maarten sent me for South African invasives. So I'm just going to talk you through that and show you some of the results. But essentially, this package is set up to be a framework that allow you to run through data that's available through GBIF, as well as local data that you might have for your own specific areas, so that you can start visualizing trait dispersion specifically and how that relates to assessing species invasiveness or site invasibility. And I'm going to explain each of those concepts to you as we go through the presentation. So just to take a step back quickly, just to give you an idea for those who haven't really worked with invasive species before, they're a major threat to biodiversity loss. And they're actually listed as one of the leading drivers of biodiversity loss worldwide. Their impacts cascade through food webs and ecosystem functioning. And understanding their establishment is key to the prevention and the mitigation of further biodiversity losses. And as you can imagine, based on that, forecasting is even more important to be proactive and to have evidence-based management. As Christina mentioned, I've been living and working in the Kruger National Park for many years, and the protected area is over 2 million hectares large. So as you can imagine, being able to forecast and proactively predict where these invasions might be impacting will enable resources to be focused into those areas rather than trying to survey 2 million hectares. But it's important to be able to predict the establishment and spread, because there's only a subset of introduced species that successfully establish. And the reason behind the establishment is a link to environmental gradients as well as related communities. And it's difficult for us to be able to make these predictions without understanding the system itself. And if one looks at a walkthrough about when an invasion happens, a species arrives at an area, does it survive or does it peter out? And it's got 3 pillars in this scenario. You've got your invaded traits, These are some species that are just built to spread. They have lots of offspring, they're easily dispersed, and they're strong competitors once they get established. Then you have the pathways and the pressures. This is how often and how many get released into a system. The more frequently organisms are moved around by ships and trucks, for example, and different trade means, the more the individuals arrive, and then the more the individuals have a higher rate of being able to make it. Then you have the habitat readiness, which even a good invader needs the right conditions. A new area needs to have a suitable climate. The disturbances like fire and land clearing or fewer natural enemies, that needs to be something that's considered. Then that makes it easier for that species to take hold. So invasions are caused by not just one thing. There are a bunch of things that need to line up for a capable species to be repeatedly introduced and welcomed into this disturbed habitat so that it can take hold and spread and cause more impacts. And how do we do this? How do we start making predictions? And many of you might be familiar with traditional species distribution models. These are models that essentially look at where a species is now and correlates that with environmental variables and predicts it into a different area. So it's essentially looking at the discovered niche and is plotting it into a new area. So it looks specifically at correlations of species occurrence with environmental variables. So it infers the ecological niche and projects that into a habitat suitability. So it assumes that these distributions reflect an environmental equilibrium. So it's something that's not changing. The species themselves are not altering the system. And it essentially then just takes those, predicts geographic suitability based on those abiotic conditions. So if you can imagine a species sitting in South America, for example, it has a specific pattern of habitat requirements and different environmental conditions that are needed for it to grow there successfully. A typical traditional species distribution model will then take those and project it into another landscape, for instance, in South Africa, and predict that those species could then also occur in that environment. So just to put this graphically, you can have a landscape. There's a site. That site has a unique combination of environmental conditions, ecological signatures or niches, if you want to call them that. And this will happen for a number of different sites. Now, each of those sites will have a number of resident species that are occurring at each of those. That then creates the signature of where that species is linked to the abiotic habitat, abiotic ecological state, what keeps that species there and what helps it grow. And that's the same for every other site as well. You can look at each of these different plants in this case. Each of them have a unique signature of the environment that they're sitting in. But you need to be cautious when you're applying these equilibrium species distribution models to dynamic invasions. So invasions in themselves, as Maarten has shown you, is something that happens over time. It's spread. And these typical species distribution models, they often ignore the resident community that they're interacting with. So they ignore what we call biotic resistance, which I'm going to talk about more in a minute. And as I've mentioned before, it assumes this equilibrium in the environment, which is rare for invaders because they're actually entering the system and altering it as they do that. And there's space for them to invade, which we will also discuss in a minute in more detail. So this risk, there's often a risk of misleading forecasts when time and this adaptation is ignored. And it may often overpredict suitability in these novel environments because it doesn't understand those parameters that are not included in the model. What you can do is if we move beyond this environment-only model we can start looking at things called functional traits. These are traits that describe ecological strategy. So a functional trait can determine, for example, growth, survival, competition. It's things like typical things like leaf area, height, or seed mass, and different things like that if we're talking about plants. And trait similarity mediates the coexistence and competitive exclusion of species. So if you can consider species living together, they would often have similar traits and they would coexist. It gets to a point, though, where those traits overlap so severely that they become competitive. And that's the basis of what the invasive map does, which I will also delve into more details later. So this mechanistic model can improve the explanatory power of a traditional species distribution model. But how do you include that? And that's the issue of linking traits to environment. And it's often called a fourth corner problem, which I'll explain in a minute. So this fourth corner problem, it's all about being able to make this connection between an environmental variable and a trait response. So we've gone through this figure previously where you've got these indigenous species that are overlaid on this landscape template. Each of these species have their own set of traits. For example, the flower color or size might attract a certain type of insect to help with pollination. And each of these, if you link it to the environmental variable, they all have a story to tell and explain or dictate why a species is growing there and how well it's doing because of that connection between its traits and the environment. So it's not only the idea behind the environment driving the species is not just because of occurrence, it's because of what's linked to that species' traits. So each of these can have their own set of traits, whether it's roots that improves the water absorption capacity, the leaf that somehow works on photosynthesis or evapotranspiration. Also, if you're looking at defense mechanisms against predators in that area, that's also something that traits seem to lean on. But if we have invasives that jump into a new environment, so this environment we've gone through in detail now, if we have new species that are introduced and they're in those settings, we can predict them based on that environment, but we also need to understand how those traits are going to drive them further. For example, an invader can be dropped off into an area. Is it going to grow there or not? And the best guess that we can make is often by drawing parallels between that invader and the resident species that are in that area at the time. And the easiest way to do that is to look at their traits. And we look at their traits in a way that we can compare them. So if we look at this I'm not sure if you can see my mouse, but on the right-hand side, you've got this trait space where you've got the gray cloud representing residents and the pink cloud representing invaders. And if you think of each of those dots in those clouds as different traits that they possess, the amount of overlap that you have between that can dictate whether or not a species is going to thrive there, but also whether or not it is going to compete with the resident species. And it's that amount of overlap that drives the decision on modeling this exercise. And that's where this invasive map R package comes in. So it's a package that's used to assess species invasiveness or trait invasiveness and site invasibility. It specifically looks at trait dispersion. So it helps us look and quantify the establishment potential at specific sites and to compare that potential across species, traits, and landscapes. It's trait-based, as I said, and it's site-specific, and it allows us to estimate this invasion fitness for candidate invasives. It assembles the resident community context that constrains this establishment and turns these quantities into mappable indicators for decision making. So the workflow is online. I'll give you a link to it in a moment, and it's also available at the end of the slide. And the example there is based on butterfly data from GBIF, but you can input your own data, and the instructions are all there. But essentially, it splits it into 3 components. The first is this functional trait space, which governs this competitive overlap that I spoke about earlier. And the trait information that the system is set up that it can retrieve it from a database called TRIES for plant species, but it's got the capacity that if you have your own trait data that you've collected on site, which is much better if you're trying to understand changes in a system locally, you can input that as well. Then it's the environmental suitability, which is the typical part of your species distribution models, which determines how well a species will perform at that site based just on the abiotic context. Then you have the biotic competition, which reduces the chance of establishment if there's a lot of competition with the resident species that are there. So this is the website that you can go to, run through the workflow there. If we have enough time, I'll quickly show you that for a second. I have got a few slides here just in case we didn't have time, so I will go through that quickly. Just to give you a breakdown of the system and how it actually works, everything is built on this idea of data cubes, which Maarten discussed earlier in his presentation. And what we do here is you have a typical site by species matrix. So you have a location. At each of those locations, you have a species, either a yes or a no, or an abundance. But linked to that site is the environmental variables as well. So this is typical of the species distribution model. And those environmental variables are linked to that specific site. Each of those species, though, has a number of traits. And those traits, because of the locality of those species, can also be linked back to the habitat. So that's that 4th corner question or 4th corner problem that I was talking about. And that's the key between making this inference of the success of species in combination with the competitiveness of residents is by these traits and that link with the habitat. So this is just a breakdown of the workflow itself. As you can see, you set up the system, you can run through a different number of internal functions that allow you to standardize the data that you've got. It specifically needs to be in a site by species matrix, so it walks you through taking your own data so that you can apply it further in the different functions. So it allows you to prep that data set. It creates this shared trait space for you. So once you've got the traits, you can put them into this into the different functions there that computes it. We can then get some outputs of what that trait space looks like, which I'm going to show you in a minute with some data that Maarten passed through to me. And then we start to do some of the modeling. So as I said to you, the model is based on the residents. So we're using the resident traits to see how well they're doing and the quantities in which they are resident in that area to help predict what are our aliens going to do. And from there, we can start making predictions about whether an invasive or invader is going to establish. Once we know that, we can start looking at how invasive is a species in terms of its predicted spread across these sites. We can take it apart into the traits as well and look at how invasive is a trait, and also based on how well it's spreading into these different areas. And then we can also take it from a site perspective and look at how invadable is a site. So this workflow is modular. As I said, it allows you to prepare these data sets, can construct these different trait spaces, can estimate this in environmental and competitive sensitivities, and at the end of the day, you estimate the invasion fitness and the establishment probability. And there's also the last function, which is a summarize function that allows you to output some variables or results that are a lot more graphical and are often used for policymakers and just for good visuals. So this is the formula. I'm going to decompose it for you so that it doesn't look like such a horrible slide. Essentially, your lambda is your invasion fitness, and it's quantifying how an invader I can grow or establish at site S. And it's taking the abiotic suitability as this environmental match that will increase the potential for growth. So the better the environment, the better the chances for growth. Then it takes the resident competition, which is going to reduce your potential for growth. And that resident competition is built on the idea of trait overlap, but also site saturation. So resident competition, how many residents are in that site, and what will the competition then related to pure numbers and space. So breaking those different parts of your formula down, you've got your abiotic suitability, which is the trait fit to the environmental conditions, because remember, we don't know the numbers of these invaders. It's one plant that's been introduced. And now we need to predict based on that environment, how well is it going to do? At the same time, those traits of the invaders, if they're very similar to a resident species, they're going to compete with them. And taken one step further again, if there are a lot of those residents in the specific site, they're also going to have a higher competitive pressure. So just to put it simply, if we look at this little diagram that we've got here, it just walks you through whether a new species can successfully establish at a site. Depends on this sort of tug of war between the environment helping it and the resident community holding it back. And those abiotic suitability is, as I said, just essentially the environmental match. If the climate conditions fit the invader's traits well enough, and then its chances to establish can rise. And the niche crowding, which is the similarity of those residents' traits, if many resident species have similar traits, they essentially do the same job, then the invader faces stronger direct competition. And so establishment goes down then, the chances of establishment will then go down. Site saturation, which is your overall crowding, even if the invader is different, a very full community with a high total competition leaves little room for that invader to actually establish and to grow. There are a number of coefficients which I'm not going to go into now, but they essentially tell us how strongly an environment helps or these different competitions hurt the invader in this site. All right. So this is data that Maarten went through. Unfortunately, he switched to Portugal, so we're looking at a map of South Africa here. But it's taking data from a Parquet setup that he had previously, and it's running it through this invasive map R package to pull out just the resident species and the invasive species. So you can see the scales on the side here are different. The colors look similar, but as you can see, there's lower richness, species richness of invasives than there are indigenous species. And if we take this conceptual graph that I showed you earlier, and we now run that actual data through the invasive map package, we see a typical trait space. So in this example, you'll see that the hull, which is this realized niche region, is marked as this block here. The white squares is the centroid, so that's where most of the residents' traits are situated. This is a PCA, so this is different axes. And then the black are your residents and your red are your invaders. So you can see where the invaders are outside of this white square. They're very, very different from any of the resident species traits. So in terms of competition with trait space, these ones outside are going to do very well, whereas the ones closer to the center are going to compete with the resident species. But as I said, that's not the only story. That's just one component of the formula. Looking at it a different way, and this is just the distance of the dissimilarity of the traits. So your invaders are your pink and your gray are your residents. So the more this overlaps, the more related the traits are. You can see there's quite a bit of a difference between them. In the example online with the butterflies, you'll see that they're a little bit more tightly related. And then running through the workflow again, you'll get outputs for each of these components. You'll get the abiotic suitability. Once again, this was based on this South Africa data set. And you'll also get site saturation, so essentially the number of residents that are filling those different sites. And then we've looked at this niche crowding already. And once you've run through that workflow, at the end of the day, you'll get your lambda, which I said is your evasion fitness. And that indicates a higher likelihood of establishment if it's positive. If it's negative, it means that it's unlikely to establish because of competition or abiotic unsuitability. We can also do some fancy footwork here and look at transforming these estimates or these results so that we can get probability estimates. And there we can start creating some of our outputs. This is just your establishment matrix. So these are all the species that were in that Parquet file that Maarten sent me. You can't see them here because we use the whole of South Africa, but these are unique site IDs. And it's basically telling you, yes, it can establish, or no, it can't establish. So everything in red means it can, everything in gray means that it can't. You can then project this onto a map as well, and you can say yes or no, is the species, for each individual species, is it going to establish here, yes or no? This is only the yes or the no view, as I said, the positive or the negative, but these probability estimates can be taken a step further, and we can look at what is that probability rather than just a yes or a no. So the same, a similar view that we had earlier, instead of the yes and no, we've now got a probability of species to establish across those different sites on the species level. And this is what we call your species invasiveness. So it essentially takes invasiveness as the property of a species, how good it is at establishing across these many different sites. So of all the sites that we projected, how many times does this species actually establish? We can take it a step further and look at traits. Okay, there we go. And we can actually look at the traits and say, all right, all of those traits that we have that we've looked at, which ones most often lead to a successful establishment? And in that way, we can start pulling out what are the traits that are most invasive. The site invasibility, similar to those black and red maps that I showed you earlier, you can now look at it at a probabilistic view. So we can plot and say how many are expected to establish in these different sites. If all the species that we modeled are predicted to establish in a site, that means that that site is very, very invasible. The site invasibility is extremely high. If very few of those actually establish there, then that is obviously lower. You can, as I said, take it through a step further and do some summaries. You can put outputs, generate some decision-ready indicators, and you can do some clustering that takes all of that data that you have and clusters your site invasibility, for example, into whether very high, medium, or very low. This is just a generalized view. There are different functions in there that you can choose for the clustering methods. the end of the day, you'll get your site and invasion fitness matrix. You'll get your establishment probability maps, your rankings of the high risk species or traits, and the vulnerable sites. And in that way, you can identify invasion hotspots and priority regions for management and for control. It also supports targeted pathways and monitoring. As I said, in large protected areas where there's limited resources, you need to try and focus your monitoring as far as you can. And it enables transparent, comparable risk assessments, which is important in any management of these invasives. This is just a quick slide. I don't expect you to be able to copy this all down now, but the slides will be made available as well as the presentation online. And so you can go through that. Email addresses are also available. So if you have any questions, you're always more than welcome to contact us directly. And also our emails are on some of this documentation here. And if there are any questions, I'm going to go to the Q&A and see if there's anything that I can respond to.
There are 2 questions open in the Q&A that are for you.
Okay.
I think the first one is, what about emerging invasive species which can establish in limited population but then come invasive after lag times? Or is establishment here synonymous with invasive?
Sorry, I didn't catch the last bit.
Or is establishment here synonymous with invasive?
Yes. So no, it's not synonymous with invasive. And you're right, not all alien species are alien invasive species. So whether it takes control of the system, that's completely different from whether it's establishing or not. So as I mentioned earlier, when it comes to spread, there's a temporal component that I have not shown here in this example. So the system, the package itself doesn't run through any examples online on how to incorporate a temporal component, which then includes the spread, which then predicts whether or not it actually becomes invasive or not. But we're still working on this package and the tutorials, and we hope to soon have a system where you can incorporate time as well. Because yes, that's a big limitation when it comes to aliens. That if you don't have spread over time, it's not going to tell you much in terms of invasiveness.
There is another one. I didn't quite understand how saturation is stabilized. Even if invasive species share similar traits with natives, if there are available resources, wouldn't they be able to coexist eventually?
Yes, and that's why the site saturation, if I can explain that a little bit more, it's to do with the number of species or the number of actual individuals. It's not about species richness. It's about the actual number of individuals. So that links directly to your resource limitations. So it's essentially a space limitation. If there are a lot of residents in that area using resources, then there is competition in that way. Whereas the niche competition is based solely on the trait overlap. So you've got them separately in the formula. One is looking at how related they are, and the other is looking at the resource limitations because there are so many individuals of the resident species that they'll need to contend with.
One very quick one. Is it possible to use my own collected data sets?
Yes, please do if you can. I would really, really love to hear how it works from your side. So the system is set up especially for local data. So I've run the example online through gbif data, but it will work really well with local data because that detail is just so much more. So if you do try it, please contact me. I'd love to hear what the output is.
I'm not sure. I'm looking at Christina. Do we still have time for one question? No? Okay. All the other questions we will answer later, and we will get back to you with the answers.
Great. Thank you. And then just a quick reminder that next week, Tsunga is going to be presenting on indicators for national invasion reporting. And then the week after that, on Friday the 13th, so I hope everyone will not be too afraid to join, there'll we'll be looking at mapping biodiversity turnover with DISMAP-R, and that will be a hands-on session, and we'll touch briefly on some of these inputs from the invasive map as well. So thank you very much.
Thank you, Sandra. Thank you, Maarten, and thank you to all the b-cubed team that helped answer the Q&As. Sorry for maybe cutting this a little bit short, but the questions just kept on piling up now at the end. But we'll make sure that we answer each and one of them, and we'll send everything, the recordings of the webinars, the slides to the participants. So we'll make all the materials available. Just to wrap up now, briefly, yes. I'm trying to quickly share my screen. Already, there has been some shout out to our next sessions. Yes, so please don't forget to join next week and the weeks after that. We'll talk a little bit more about the biological invasions. And thank you again for these amazing talks where we learned about these repeatable and freely available workflows that can be customized depending on our research interests. There are additional resources that can be used by the participants available on the project website under the guides and tutorials tab on top of all the links that Maarten and Sandra made available for us. And of course, you have their email addresses, and if you have additional questions, or if you want to receive a certificate of attendance, this can be made available upon request. Just contact us using this info@b-cubed.eu. There are a lot of social platforms that we are active on. Aside from the website project, you can find us on different platforms, and of course you can sign up for the b-cubed newsletter. Make sure you spread the word, and hopefully you enjoy this time spent together. You've learned. I certainly learned a lot. So thanks again to all speakers, all b-cubed team, and hopefully we'll see you next week as well. Same time, same place. So thank you. Enjoy your weekend, and thanks.